Quality control and biosafety and tissue engineering are essential pillars for ensuring that biofabricated products are safe, effective, and reproducible, especially considering their direct application in patients. Quality in these processes involves obtaining accurate and reliable results by measuring predefined specifications, including parameters for safety, identity, purity, and biological potency. Likewise, the aseptic process is of central importance, considering viral and microbiological safety throughout the entire manufacturing chain. Concepts such as sterilization, disinfection, and asepsis must be integrated into validated protocols that include mandatory sterility tests, endotoxin studies using in vitro techniques such as the LAL assay, mycoplasma detection, preferably by PCR, and assessment of the risk of viral contamination, both endogenous and adventitious. In addition, aseptic process simulation tests allow for validation of the overall integrity of the production procedure. Furthermore, the transition from basic research to clinical production requires the design of cell production units that adhere to good manufacturing practices, GMP, where everything is protocolized, planned, and documented. Together, quality control and biosafety not only protect the patient but also ensure the traceability, standardization, and regulatory viability of tissue-engineered products in a highly demanding clinical environment.